Support Microbe TV. It's time for our annual fundraising drive. We depend on your support to produce high-quality science videos and podcasts, and now is the time to help us. If our programs have appealed to your science interests, or if they have helped you in some way, please record a brief audio or video with your phone and tell us about it. Let us know how we empower your inner scientist. We'll use it for our fundraising efforts. Send it to incubator@microbe.tv. And for more information on how you can give us your support, go to microbe.tv/contribute. This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 1175. I'm Vincent Racaniello. You're listening to the podcast all about viruses. Joining me today from Western Massachusetts, Alan Dove.
Good to be here. It's 31 Fahrenheit, -1 C, and breezy, and we actually got the first snows of the season recently. It was only an inch or two. But it's properly winter here.
It's 1 degree here in Chelsea. I just want you to know. See, it says Chelsea, Alan.
Oh, nice.
Chelsea.
Right down to the neighborhood.
It's really the edge because it's 30th Street, which is
just in case there's different weather in Tribeca. Yeah.
There actually could very well be.
Right? Yeah.
Microclimates. Also joining us from College Station, Texas, Jolene Ramsey.
Howdy. Here in College Station, it's 55 degrees Fahrenheit and 13 C. It's sunny outside, but it's windy, so it feels cold. And it is cold for Texas.
For Texas, yes.
No sympathy. No sympathy. I mean, I wait for the train. It's bitter. It hurts. It hurts. Yeah, I would try walking up and down the platform to stay warm, but there's no place to stay inside. Yeah, it's really cold. Yeah, but next week, I think it's going to warm up. Today, it's all low. Tomorrow, -4. Oh, Sunday, 1 degree. Monday, 1 degree. Tuesday, 5 degrees. Balmy. Wednesday, 6. But then Thursday, -1. Friday, -2.
These are your highs, Vincent, right?
Yeah.
Those are Celsius.
Yes.
For Celsius, of course.
Just to be clear.
But just to let you know, the air quality is good.
Yeah. Wintertime is great for air quality.
And the humidity is very low.
Yeah.
And that's why respiratory viruses, one of the reasons why they transmit well in the winter, it's cold, low humidity. Good conditions for transmission. If you enjoy these programs, we'd like to have your support. In fact, we are in the fundraising part of the year, November and December. Go to microbe.tv/contribute. A number of people sent in little recordings, audio or video, about what Microbe TV means to them. We've been putting them up slowly on Instagram and YouTube.
I've been watching those on YouTube. It's in my news feed. They're good.
We got one from a freshman in Shanghai. It was so unexpected. Hi, I'm a freshman in Shanghai. I love TWiV. It was great. Yeah, many people. We asked people to tell us how you found your inner scientist. And Betty from Washington Heights. Can you imagine, Alan? There's a lady who listens.
Right in Washington Heights.
She lives in Washington Heights. It's because Alan and I used to be there. That's where Columbia is. She said, I didn't even know I had an inner scientist until I started listening. I love to hear these informal. So you can send one in too. There's I think there's instruction. Yeah, at microbe.tv/contribute, there are instructions. You can email it, or there's also a Dropbox that we set up as well. And also remember to write to your senators to oppose nomination of everybody, basically, because they're all hacks. But RFK Jr. as head of Health and Human Services seems to be the worst. Although I went over on the live stream this Wednesday. NIH, CDC, FDA, Medicare and Medicaid services, Surgeon General. I mean, they're all just flawed.
Well, and that's just our little corner. I mean, if you look at his nominees for national intelligence and everything else, it's just I'm waiting for him to find some cocaine dealer he can pardon and put in charge of the DEA. It's just, but it's all stunt programming. I mean, this is all being done for shock value to make you look. And some of these are going to go through.
Yeah, we can't oppose all the science ones, but RFK is really bad. SafeCommunitiesCoalition.org/RFK. Let me see if it works. Someone told me it was wrong. Yeah, that's it. Safe Communities Coalition, it's all one word, .org/RFK. Now we have some announcements. First of all, a job announcement to release the viral immunologist in you. I didn't know I had a viral.
It sounds like a great idea.
Yeah.
Rosenfeld Lab in the Division of Viral Products at CBER FDA is looking for a postdoc staff scientist to continue the lab's research, understanding the immune responses elicited during enterovirus infection. If you're an immunologist, microbiologist, or virologist wanting to learn more viral immunology, email Amy at amy.rosenfeld@fda.hhs.gov. Notice the email, fda.hhs. So FDA is within the HHS, right? So RFK Jr. supposedly would be the head of HHS. So
HHS it's the top level of all of the health and biomedical research, most of the biomedical research stuff in the country.
And he nominated a lawyer who knows no science whatsoever.
Of course.
I think you would do a better job, Alan.
I couldn't probably do worse.
In fact, in your own honesty, you would say, I'm not qualified for this, right?
Yeah, no, I would say I'm not the guy for this.
Even though you have more science knowledge than he does.
Sure.
Oh, my gosh. Okay, amy.rosenfeld@fda.hhs.gov.
And by the way, HHS is also like Center for Medicare and Medicaid.
Right.
So this is if you care at all about health insurance or any of these other, even if you're not particularly in the biomedical research community, it's the medicine agency.
And you know who we nominated for that? The head of that, right?
Yeah.
Dr. Oz.
Mm-hmm.
The TV personality who bunch of
he actually, he's kind of gone the way of being a showman, but he is actually a doctor who has accomplished a lot as a physician. And
yes, but now as a TV
personality. Yeah, he's just a showman.
He hawks like snake oil.
Yeah.
For weight loss. He's on record saying, I could sell a cancer treatment every day of my life. People will buy anything.
Yeah.
It's terrible. The doc, you're supposed to take a Hippocratic oath and not do any harm, right? Anyway, let's go on to ASV. ASV, American Society for Virology 2025. It's going to be at in Montreal, at the Palais de Congrès de Montreal, July 14th through 17th. It will be warm there. I'm looking forward to it. ASV.org/ASV2025. You should renew your membership. It goes from January to December, or make a new one. It's not very expensive for what you get. And you need the membership for travel awards, for ASV cares, and on-site child care. If you're no longer a trainee, membership gives you discounted registration. So you go to the meeting site, click on grants and awards, travel awards, and then on the 5th line down, it talks about membership. Or you can find it on the main ASV website, asv.org. Okay, this one is entitled Poliovirus' European Vacation. Actually, it's not the article title, but it's just what we wrote here.
The World Health Organization does not go for clever headlines, but this is a good name for it.
This is from the WHO. Poliovirus detection in European region underscores the importance of vaccination and vigilance. So poliovirus has been detected in wastewater systems in 3 countries, Germany, Poland, and Spain. And this is circulating vaccine-derived type 2. And these countries all use inactivated poliovirus vaccine. They have high rates of immunization. So there's no cases of poliomyelitis. They just find the nucleic acid in the sewage, which means someone is excreting it somewhere in those countries. And it's the same in the U.S. And if you let vaccination rates drop, someone's going to get polio.
But they do say that these 3 countries where they detected it have 85 to 93% coverage nationally.
Yep.
Which is high, but maybe it needs to be higher.
Maybe closer to 100 would be good. Yes.
Yeah, I forgot the number, but high 90s. So here in the U.S., it's mostly over 90%. And in Rockland County, a couple of summers ago, it dropped to like 30% in a certain age group, and boom, we had a case of polio there. So you have to be vigilant. Now, I'm sure we have poliovirus in sewage in the U.S., but the CDC doesn't look for it. They don't want you to know that it's
Because,
there.
well, they don't want to know. That's,
they don't know
see
what
no
they're
evil.
gonna do.
I think the wastewater treatment plants, depending on where you are, some of them are very amenable to allowing people who come for research purposes to take samples and look at things, and others are not so amenable because they're afraid of what you might find. So we do a lot of wastewater sampling to do phage hunts, and we just know that some of the wastewater treatment plants are very friendly, as in like they almost don't even notice if you walk up and take some, and they're like, oh, hey, see you later. But others, they are literally either they completely do not respond, or they finally respond and say, that is against our policy. We cannot allow you to come here. We don't give out samples, and so on and so forth. So I imagine that there are some places where this kind of surveillance could be done here in the U.S., but it will be highly dependent on the people who run.
On jurisdiction. And just let that sink in for a moment, everybody. There are sewage treatment plants that will refuse to give away sewage to researchers.
I was in New Orleans a couple of years ago, and I talked to a graduate student who they were doing wastewater sampling before and after Mardi Gras. They wanted to see the STDs, basically, how they go up before and after. So the bacteria that are associated with STDs, right? And he said, I have these samples. Maybe we could look for polio. I said, yeah, you should. You could just design a PCR. He made the mistake of asking the CDC for permission, and they said no, of course.
Mm-hmm.
No, it's better to get forgiveness than permission. You just do the experiment. Yeah.
The problem is many state health departments, they're dependent on financing from CDC, so they don't want to get them mad at them. So, all right, next thing is this is interesting. As you know, there's a mystery disease in the DRC. Killed 143 people so far. It's a respiratory disease. They have flu-like symptoms, high fever, severe headaches. Mostly women and children are dying. And this is many days now. It's like since Tuesday or Monday, I think. And I suppose the state of the system in DRC is that there are no labs to do PCR.
Yeah, so
right?
this area, so this is in the southwest of DRC, in a very rural province, and the DRC itself is like a lot of mountainous jungle, and the infrastructure has been progressively destroyed by everything from colonialism onward. And this actually, I saw another article on this that said that the first cases of this were like 6 or 8 weeks ago. And it took a while, first of all, for the local health authorities to see that there was this unusual thing going on, because against the background of other diseases in the area. And then it took a while to get the message to the capital, and then for the authorities to actually get involved. So it's taken a while to get to the point of saying, hey, there's actually maybe something going on here. And this is a very poor area. There's a lot of malnutrition. There could be a number of causes for this, or it could be some new virus emergence. We don't know.
Yep. And I mean, they're sending samples out. It is not hard to do. You take a nasopharyngeal swab and and do a PCR using a panel of primers
Yeah.
against respiratory pathogens, right?
Once you actually get the sample and you're in a 21st century modern lab, not
That's
hard
what
to
they
do.
did in Wuhan in December 2019, right? They saw all these unusual pneumonia cases, and within a few days, the doctors in the hospitals said, "Hey, this is different," and they looked for all known bacteria and viruses, and none of them were there. So then they sequenced it, and boom, new virus.
And that's was happened quickly because it was happening in the middle of a major city with lots of government presence to connect all this stuff. And
okay, and finally we have BMG. Did you put this in, Alan, this time?
I don't think. Okay. No, I didn't put this in. Was this you, Jolene?
I think it must have been Amy.
Ah.
Okay. It's a BMJ journal, British Medical Journal. Global Health and Economic Burden of Chikungunya from 2011 to 2020. Basically, they analyze the global burden of chick and calculate disability-adjusted life years, dailies. It's all computational using data. The outcomes were number of cases, the total daily burden, and the direct and indirect costs of acute and chronic chikungunya. So chikungunya virus will cause an arthritis-like syndrome, joint pain, and in many people it persists for a long time, and they can be debilitated, right? So they can't work. So they found 18.7 million cases in 110 countries, causing almost 2 million dailies, right? Disability-adjusted life years. Most of it in Latin America and the Caribbean. Total economic burden, $2.8 billion in direct costs and $47 billion in indirect costs. Long-term chronic illness was one of the biggest source of costs, right? People have to be treated for chronic illnesses.
So chikungunya, is one of those mosquito-borne viruses, and the acute phase on its own is pretty bad. Chick has been known since the 50s, but it only in the past couple of decades became more prominent, and we had more cases. And it wasn't there are other alpha viruses that have similar symptoms, but here now there is actually a vaccine for chikungunya virus. It's an attenuated vaccine, but I don't think it's relatively new. It's only been approved for about a year here in the U.S. anyway. So
I know that chick is acquired locally in Texas.
Yeah.
Right. As opposed to being imported. Many of the cases in the U.S. are travel related, but in Texas it's locally. So I wanted to see the state of that. So I Googled local chick Texas. I got all the chick Wrong term.
Chick V.
It's like when I was shopping for darkroom equipment on eBay and I searched for enlarger. No. Photographic enlarger.
So it's transmitted locally in Texas. I think also in Florida.
In Florida, yeah.
Because you guys have the right mosquitoes, Texas, and the right temperature. Anyway, so this chick has a higher burden than we thought previously. So maybe the vaccine needs to be more widely deployed.
Mm-hmm. Yeah, it's like I said, it's relatively new, and chikungunya is not as well recognized as something like dengue or yellow fever that are also in these same areas where those mosquitoes frequent. So I used to study a relative of chikungunya virus, but I didn't know about the vaccine until one of my colleagues told me about it recently. So I think that maybe more information for those who are traveling to areas or live in areas where chikungunya is a risk, because the acute disease isn't great, as I've heard from friends or colleagues who lived in places where it was endemic that came and started working with us. But the longer term effects are definitely a real problem.
Chikungunya means to become contorted.
Yeah.
In the Kimakonde language. Do you know where that's from? Kimakonde?
It's somewhere in Africa.
It sounds like it, yes.
Kimakonde, an ethnic group in northern Mozambique and southeast Tanzania. Okay. Yeah, it's Africa. Okie dokie. By the way, chikungunya is a great name, isn't it? A great word. It used to be mainly vectored by Aedes aegypti, and as Julian says, we've known since a long time. And then in 2000, a variant of chick was selected that had a single amino acid change in the spike protein, which could now reproduce very well in Aedes albopictus, a different mosquito, which is more broadly located. And Aedes albopictus is in much of the U.S. and other parts of the world, so it's expanded its range as a consequence. Aedes albopictus never used to be in the United States before the 80s. It was brought into Houston in the used tire trade. Mm-hmm. Water in the tires, you can't get rid of them. If someone could invent a way of keeping water out of used tires, they would be wealthy.
A cost-effective way. These are bulk freight that's got very rotating. Low value, and so yeah,
there's no cost-effective way. Maybe just covering but then there's water in them. That's the problem.
Yeah,
can't get rid of them unless you have someone take each tire and tip out the water. Right? Not going to happen. Okay, we do have some papers for you today. The first one is snippet. They're both really snippets, so it doesn't really matter. But this one is Nature Microbiology. Too bad Dixon isn't here.
I know.
Yeah, he'd be handy to have for this.
Diverse RNA viruses of parasitic nematodes can elicit antibody responses in vertebrate hosts. Look, Yella is on this. So Yella hosted a meeting in Belgium years ago, double-stranded RNA virus meeting, and I learned how to pronounce his last name. His first name is Jelle, and most people would say Jelly, but it's Yella. And Matinj, I don't know how to pronounce it. I forgot. If you don't pronounce something over
Matinsons would be my attempt.
Matinsons, that could be. So the authors are Shannon Queck, Hatterman, Wu, DeConnick, Hegd, Boucher, Cresswell, Foreman, Stephen, Lacourse, Ward, Wanji, Hughes, Patterson, Wagstaff, Turner, Parry, Cole, Hines, Otabil, Matinsons, Cole Bunders, and Mark Taylor. And in a practice I really don't like, they put the affiliations at the end of the paper. At the
end of the paper.
Is not podcast friendly.
No.
Okay?
It's not even reader friendly. If you're just casually reading the paper and say, where's this group from? Oh, flip, or scroll, and you got to go past the citations and everything.
All the way at the end, just before the extended data. So this is from a bunch of different groups. Liverpool School of Tropical Medicine, University of Antwerp, KU Leuven, University of Bouea, which is in Cameroon, Brock University in Ontario, University of Queensland, which is in Brisbane, and the University of Energy and Natural Resources in Ghana. Okay. Yeah, it's now I have to scroll back up. Scrolling back up is easier. That's fine. So the title says parasitic nematodes. Nematode is a worm, and a worm of different shapes and sizes. They can be round, they can be flat, they could be tiny and look like hair. I've learned a lot about this from Dixon on TWiV. In fact, the first 21 episodes of TWiV, he just went through all the parasites. And so he went through I used to say, Dixon, is it nematode or nematode? So whatever you prefer.
This is a hugely important group of parasites worldwide. That cause all kinds of different problems in probably billions of people.
And also animals, agriculture.
Yeah, and animals, and they encompass some of the oldest pathogens that have infected humans and all branches of life. And
yeah, as it says here, over 1 billion.
Well, not all branches of life. They multicellular life. So.
over 1 billion people. And we just learned about dailies, 7.5 million dailies globally. So some examples you should, if you listen to TWiV, you'll know. Ascaris lumbricoides. So that's the giant intestinal roundworm. It's huge. Oh my gosh. And it can live in your intestine. And when it comes out, you're like, that was in there. Trichuris trichiura. And then hookworms, Necator americanus, Ancylostoma duodenale, and Ancylostoma ceylanicum. Okay, so those are some. And then we have, those are intestinal. Okay. And then we have some others like the filarial nematodes, Wucheria bancrofti and Brugia malai. Onchocerca volvulus, this is the most famous. This is the Jimmy Carter parasite. Right? He's trying to eliminate it in Africa. Well, he was. He can't do anything anymore, but yeah, he's over 100, right? So that causes river blindness, right? That's Onchocerca volvulus. And then, so there are lots of these nematodes. In this paper, they want to know, are there viruses in these nematodes? We know that some parasites...
Doris used to work on these, right? Some of these. Didn't she at some point?
Let me see here. She used to work on ivermectin.
Right.
Oh, Onchocerca, right.
Yeah, Onchocerca, yeah.
Yep.
Anyway, yeah.
Connections.
So in this paper,
I just was the word ivermectin. Someone sent an email. Apparently, someone has published a list of all the good things that ivermectin does, like 20 bullet points. Every one of them is fabricated.
Wow.
But how would a normal person know that, right? How would a normal person? They want Daniel to comment on it. He's just going to say they're all BS.
Mm-hmm.
So what ivermectin can do is help with Onchocerca volvulus. And then, so we do know that there are viruses of parasites, and we've covered some of them on our podcast. Plasmodium have viruses. We just did the last TWiP on that. Trichomonas vaginalis and Cryptosporidium parvum have viruses as well. Leishmania. And these are typically RNA viruses. And what's interesting is they make the human infection worse. And the idea is that the RNA is sensed by the innate immune system, and then it causes more inflammation than you would normally get without the virus. So this paper, actually, both papers today are about discovery, right?
Mm-hmm.
Can you discover viruses? So what their approach is, they take transcriptomes. So transcriptome is you take one of these nematodes, you grind it up, you extract the RNA, and you sequence all the RNA. And the transcriptome refers to that, a messenger RNA, right? That's a transcriptome. And so they have 41 nematode species, both human and animal, and they take the data, which other people have generated, the sequence data, and they analyze it themselves. And they find 91 virus or virus-like genomic sequences across 28 species. And they characterize 2 of them in some detail and provide some evidence that they actually exist as viruses. Because remember, when you find a sequence, it's a viral genome. It's not a virus, right?
And we're all full of viral genomes.
We're full of viral genomes. The question is there a virus? And some people say, of course, if the genome is there. Well, no, not necessarily. It could just be persisting. But they provide some nice evidence that these things may be replicated.
I think it's real important to emphasize that these are worms, and worms they're not huge in and of themselves, but they have their own microbiome. So they are colonized by microbes, including bacteria and viruses, and possibly the viruses that infect those bacteria, maybe fungi. I don't know. I've not heard of archaea in worms, but it's possible. We have them in our microbiome. So when they grind up the worms and pull out all the RNA, in this study, they're looking for viral RNA, but it's RNA from the worm and RNA from every other organism that's there. And it could also be RNA from things that they ate.
Yeah. Okay. So they mine these transcriptomes, publicly available, so anybody could do this. Elon Musk could do it.
At least the first part of this.
41 species of parasitic nematodes, and they find 91 virus or virus-like sequences. And the way they're looking for this is they look for the RNA-dependent RNA polymerase, which is the most conserved protein in RNA viruses. So you can use that as a hallmark. Someone asked me the other day, what's the most conserved human protein? Going all the way back to Neanderthals and Denisovans and Homo sapiens and so forth. You guys know what that would be?
That's distinct to humans?
No, just the most conserved, like the RNA polymerase of RNA
ribosomal
virus.
genes are a classic, like everything's got them and they don't vary
It's
much.
not the most conserved human protein.
No. Oh, okay.
It's actually a histone.
Okay.
Which makes sense, right? It's got to bind DNA.
I don't know. That doesn't really make sense to me, Vincent, because everything has DNA.
Everything has histones, right? Yeah.
What do you mean?
But they're used a lot in gene regulation, so they would vary more, maybe?
No, it's just in humans. I mean, it may be in other animals as well, but
worms have histones too.
If you look at all the humans on the planet, plus all the sequences that we have from Neanderthals and Denisovans.
So you're saying they all have histones?
They're highly conserved. Yeah, it's the most conserved out of all the proteins that you can get out of the genome sequence. Right, the histone actually is a specific histone H4. It's the most conserved.
So as Bob Dylan would say, everybody must histone.
Indeed. Okay, so these viruses were from 24 families. Right, family is like Picornaviridae with the funny letters at the end. That's a family. So there's a lot of diversity there. And then I'll tell you what the common families were, but a lot of them not going to ring any bells. Totiviridae, there were 36 genomes. Partitiviridae, 8 genomes. Rhabdoviridae, that should ring a bell. And Totiviridae, 36 genomes. Lispiviridae, 6 genomes. All right, so they find lots of sequences here. Okay, so then they find a few of those are contaminants. 3 of them turns out to be bacteriophages. This is going to ring a bell in the next paper.
Does happen. They're everywhere.
Yeah. And so they just work with the rest. So then they take all these sequences and they compare them to what's available on the database, the NCBI database, to see if any of these had been previously identified. And they have been, right? 3 of the 4 viruses in Ascaris had been isolated previously from pigs in the U.S. From this study, the Ascaris were from pigs in the U.S. They were previously identified from parasites isolated from pigs in China. And the names of these viruses, I want to tell you because I like them. There's the Hubei rhabdo-like virus, the Hubei toti-like virus, number 12, and this is my favorite, the Wuhan large pig roundworm virus. A large pig.
No, I think the virus was large. The pig was probably normal size. That's my guess.
I was going to ask you, what is the large in the right place then?
Yeah.
I like the name anyway. So basically these Ascaris viruses are widely distributed. And then they say there was another study in Brazil that identified the Wuhan large pig roundworm virus 1 in human fecal samples, so maybe this can infect people as well. Or people do have roundworms, so maybe that's where it came from. And then they were able to assemble some complete genome sequences for previously published virus, so they can see what kind of virus it is. The first one, for example, it's a unnamed virus, and the second one is an RNA virus in Toxocara the dog tapeworm, related to the Jingmeng virus group, which we've talked about a long time ago here. These are related to the Flaviviridae. And as they say, the first Jingmeng virus was identified in ticks, and then subsequently in Toxocara canis. And so with these exceptions, there are like 3 exceptions. None of the other viruses, 86 sequences, were in existing databases. So they're all novel sequences, which is very good. Must make them feel good. Can you imagine all 91 had been previously discovered? Yeah.
Or all 91 being novel would be a little, what did we miss?
Yeah.
Mm-hmm.
So then they focus, the rest of the paper, they focus on two viruses of Brugia malayi and Onchocerca volvulus. All right. And they found 7 RNA genome sequences in these two, 1 in Brugia and 6 in Onchocerca. And the one in the Onchocerca was confirmed in experiments on adult worms collected in Cameroon and Ghana. So they actually went to those worms and redid the sequencing, and they find the viral sequences there. So that's confirmation of that.
And I'm
So-
assuming a reason for picking these particular worm species is that they're relatively easy to work with in the laboratory?
It could be. Maybe because they got complete sequences. I don't know. But maybe
Yeah, that too.
the worms you can work on, yeah. So Brugia malayi RNA virus 1, it's a toga virus. It's actually an alpha virus within the toga virus family, which is what chikungunya is. It's an alpha virus within the toga virus family.
Yeah, but I was really surprised. There is diversity within the toga viruses, but it's missing all the genes for making envelope proteins.
Yeah. Yeah, they do have a figure of it here. Yep.
Yeah. It has all the non-structural region.
Mm-hmm.
I mean, it's recognizable as an alpha virus because it has the non-structural genes, which are made as a polyprotein, followed by the structural genes, which are made under a subgenomic RNA. That includes the capsid that comes first. And then after that, there should be envelope proteins.
Yeah, maybe is there sequence there, or are they missing it?
They say that it's like a 7 kb genome, which is too small to include the envelope proteins.
Yeah, that's right. It should be around 11, right?
Yeah.
All right. To be sorted out.
I just thought that was, it's interesting. Maybe this is like a very different kind of, very different branch of the toga viruses.
Yeah. All right. And then the Onchocerca sequences. So there are Onchocerca volvulus RNA viruses 1 and 2. There are rhabdoviruses and disruptive. So 2 different virus families. So rhabdoviruses, of course, include rabies virus. And the disruptive, these are plus-stranded RNA viruses. They're unusual. They have 2 open reading frames. That's the disruptive, right? With an internal ribosome entry site in between the 2 of them, which is how they get translated. So they looked at, so these parasites exist in different life stages, right? It's not like a virus that is just one virus particle, right? These things have filaria and microfilaria and adult, juveniles, all kinds of different stages. Dixon could go on and on.
Oh,
Dixon,
yeah.
He'd be talking about it for a long time. But they look at the sequence reads. In other words, when you do transcriptomics, they do the sequencing from different life stages, and then you can count the number of reads per stage. You can get an idea of the relative abundance. And this Brugia virus is in all the life stages of Brugia malayi. Don't ask me what they are because I don't remember. But again, Dixon could just knock them off right here. I do have parasitology textbook here, and it has wonderful figures which would tell you all the life cycle.
Well, another thing that they took care to check is male versus female abundance. Yeah. And they did see that the virus was present in both, as well as in the adults. But in the adults, they saw differential abundance in male versus female.
Yeah. And for the ORV, the Onchocerca, they don't find it everywhere. So they say it may not be ubiquitous in all parasites. And they also looked at life stages. So here they take microfilaria from Brugia malayi. They do RT-PCR on it, and they can see that you actually amplify. So if you take worms that you get separately from this study and you do PCR, you can see the viral sequences being amplified. So it looks like those sequences are in those worms. Yeah, all the regions amplified to the expected sizes. And they do that quantitative PCR from different life stages, and it varies. So the larvae have low copy numbers, for example, and I'm not going to go through it because it's not going to make a lot of sense. But there are differences in the different stages of the parasites.
And both the papers we're talking about today are open access, so people can go look into that.
Then they did something interesting. They said, let's look for small interfering RNAs in these hosts directed against these viral sequences. Because we know from other studies that when RNA viruses infect hosts, the host can often make interfering RNA by chopping up the viral RNA and using it as a defense. So they look for small RNAs against these viral sequences, and they find them 23 nucleotides long, which is the right size. They find for the Brugia malaria virus, they find these small interfering RNAs, which suggests that these might be replicating in those hosts, right? So that's cool. So then they make antibodies against. So this is good. They're doing a lot. They're not just doing bioinformatics.
It's a thorough paper.
Make antibodies against. So they have to take the gene for the capsid protein of Brugia malaria virus, and they produce the capsid protein in recombinant fashion. And they inoculate rabbits with the protein. They make antibodies. And they can do Western blots of, in this case, they do microfilaria and adults. And they find proteins. They find the capsid proteins in the microfilaria and the adults, which that's interesting, right? It further suggests that these viruses are actually reproducing.
They're real.
Because
yeah.
The capsid proteins are being made. They can also find the proteins in the culture medium. So if they grow the nematodes overnight and they take the culture medium, they can find the capsid protein in it, suggesting that maybe it's being released. They could do electron microscopy of that medium. That would be cool. Or the parasites themselves. That would be very cool to show particles. But maybe that was beyond the scope of this study. That's what you write when the grant reviewers say you should do this, and it's beyond the scope of this.
Well, they did check RNA.
They checked RNA, yes. You're talking about the fluorescent in situ hybridization.
Mm-hmm. It's beautiful.
Experiment.
Yeah.
Yeah. And so you can make RNA, which is fluorescently labeled, and then you can hybridize that to the, in this case, the parasites. And then you do microscopy, and you have very beautiful images, different colors. And you can see that, wait a minute, I just lost my place. Oh, here we go. So this is called FISH, fluorescence in situ hybridization. And they say most microfilaria show a weak signal for virus RNA. Adult males and females have intense signals in patches just below the nematode cuticle. And in older nematodes, it's more patchy. So you can see, so this is looking for the genome in these live worms, and it's there, which is cool. It's not, and it's separate from the worms used for the transcriptomics, obviously, because that was done by someone else.
Right, and it's not everywhere. So
no, it's not.
That kind of localization, it's not like it's just in the gut. And
right.
It's if it were replicating in tissue-specific manner, this is the sort of thing you would expect if it were actually a worm virus.
Right. Then they took Onchocerca worms and they did RT-PCR and Western blots to see if the genome and the proteins were there. And in fact, RT-PCR is positive. They can amplify the viral RNA in, again, these are worms collected, in this case, from Guatemala and Cameroon and Mali. Different places, they all have this virus in it. Very interesting, right? Because you can imagine if it were only in one place, that would be localized, but it's apparently more widespread. And then the Western blots confirm this. they can find. So these are different antibodies. This is against the viral glycoprotein. Is that the one that you were saying isn't present, Jolene?
Yeah, but this is the other virus. So
It's the other virus.
Yeah.
It's not Brugia. It's Onchocerca.
They
Okay.
looked at capsid in the Brugia virus and
Right.
Yeah.
glycoprotein in the other one,
Yeah.
the Onchocerca.
So they make antibodies against the Onchocerca glycoprotein, and they can find the protein in different stages. Infective third stage larvae, adults and so forth. So the genome and the proteins are present in field isolates, let's call them. That's pretty cool. They also do immunofluorescence localization of Onchocerca virus using their antibodies. And they say the most commonly stained tissue is the epithelial lining of the female reproductive tract. And they get signals elsewhere. But they all the nematodes they looked at 6 stained positive for the glycoprotein. So they're all presumably infected. I think there's pretty good evidence that they're infected. Then they say the people make antibodies against these viruses, right? So they make recombinant proteins from the Brugia and the Onchocerca, the capsid and the glycoprotein. And then they can use that recombinant protein to look for antibodies in hosts. And their control is the Mongolian jerd, which is used to grow.
I had to look this up.
Yeah, I did too. It's a gerbil.
Exactly. It's cute. It has these dark paws.
Yeah.
So that's used for growing the Brugia virus, the Brugia malayi. So they're going to use it as a control. And then they have serum from humans who have been infected or exposed to Onchocerca. These are sera that you can get from collections. So then they do Western, but 7 of the 9 jurors infected with B. malayi were seropositive for antibodies. So that's their control. So their antibodies are okay. And they also do ELISA as well as Western blots. And in the ELISA experiment, and they do that on jureds and humans, 11 of 12 jureds were seropositive compared with uninfected control. And all of the humans who were exposed to ovovolus were seropositive. So these people all had been exposed to
So the human the human data are a little bit split. So all the human samples from sub-Saharan Africa in Cameroon, Nigeria, and Togo were seropositive. Uganda, all but 4 of them were seropositive.
Yeah.
And then in Ecuador, only 48% were seropositive. So there's something going on with the difference there. Maybe.
Yeah, but it's interesting. Some humans are seropositive.
Yeah.
So they're seeing the virus.
Most humans are seropositive.
And making antibodies against it, right? And I presume that's because they have the worm and
So the people have an infection with these parasitic worms.
Yeah.
The worms are infected with the virus. The virus makes its proteins that are somehow exposed to the human body, and the human immune response makes antibodies against those worm virus proteins.
Mm-hmm. So that's the data. I think it's for these 2 viruses, the Onchocerca and the Brugia, it's good evidence that they exist as viruses. And it looks like, gosh, they're in all parasites that you collect. Well, they didn't use huge numbers, right? But I don't see an uninfected parasite. Right?
These seem to be very widespread infections that are probably ancient in these parasites because of the global distribution of them. It suggests that this was going on before the worms even spread with humans around the world. I mean, the one thing that would be beautiful to see here, which may not be possible simply because all the worms are already infected, is to take uninfected worms
Yeah.
and see if
Try
you can
infecting
infect them.
yeah.
What they
But-
did say at the beginning was that 70% showed evidence for these 91 sequences. So only 28 out of 41, I thought.
Right. No, I was talking about the BMRV and OVRV.
Mm.
If you
Ruby
could
and
find
the-
uninfected
Well,
worms.
Maybe you
presumably,
could,
if they didn't have any virus-like sequences, they're not infected.
That would, yeah.
Presumably. Maybe they could remove the viruses from these worms. I don't know how, but
Yeah. I'm just thinking, could you fulfill Koch's postulates with this? And
yeah, I think it would be useful.
probably
to
pretty
infect.
challenging to do, but
But maybe you could treat the worms with their antibodies and get rid of the virus infection.
immunotherapy on the worms and then,
Yes, immunotherapy.
RNAi.
I think that
RNAi.
RNA. Oh, yeah.
we could do RNAi, yeah. I mean, the hosts are already doing RNAi, right? But apparently it's not working. But maybe you could help it.
Ramp it
But
up.
I think that's an important experiment. It depends what you want to do. If you want to study, I mean, you want to know what the virus is doing, right? If it's harming the parasite, if it's helping it, if it has any effect on disease, right? And you need to have an uninfected control to do all
well,
those experiments.
I don't think that they would infect a human, but I guess you could do it in the GERD.
In the GERD.
I mean, you need a parasite with
Yeah.
and without virus, right? Yeah, you could use gerds. I don't know if they get disease or not. But apparently, some of these humans have been exposed. They say, ovolvulus infected or exposed. I don't know what that means. I don't think you can infect people with this. This causes blindness, right?
Yeah,
Peripheral
no, you
blindness.
wouldn't.
You don't want to do that. But, any kind of pathogenesis you want to do, you need to have an uninfected parasite.
Right.
Without virus, right? Because I think we'd like to know what these viruses are doing.
Oh,
Okay.
exposed individuals may be people who were treated for these diseases.
I don't know.
So they're not currently infected, but they were previously infected.
Maybe, yeah.
And since you're doing a serosurvey, they would still have antibodies.
So they have, they've got viruses of human parasites and also livestock. There are a bunch of them that we haven't talked about, and I can't pronounce anyway. Trichinella, that's the one I can pronounce. Dixon's parasite. They found, virus Trichinella infect livestock, and they find viruses there. 37 unreported viruses of parasites that infect cattle, sheep, and pigs. So many animals harbor Trichinella, folks, like bears and deer. So you shouldn't eat raw bear meat.
No.
In fact, we did a paper on TWiV a couple of weeks ago where there was an outbreak of trichinellosis, I think in Arizona or Colorado. These people had eaten kind of rare bear meat. And many of them got sick. And the bear meat is very red, so they couldn't tell if it was cooked enough. And they ended up eating it too raw. But, you need to use a thermometer, folks, right?
Yep.
But don't eat bear meat because bears have trichinella. And if the worst
Well,
thing...
I mean,
But
just
if you-
cook it.
if
Cook
you
it like pork.
But if you freeze it, then that's okay, right? Freeze it first.
freezing it should be okay.
and
I'm
then
not
cook
sure,
it.
actually. It's a good question.
That's
Yeah,
that's
cook-
my recollection from what
maybe
Dixon
that's
has
right,
said.
Yeah,
Yeah.
freezing kills it.
Yeah.
Freezing for a period of time, and cooking thoroughly. And
Right.
and I would say if you're going to eat bear, just cook it the way you would pork.
And folks, if you hunt and bear, deer, whatever, don't feed raw meat to your dog because you could give your dog
No,
trichinella.
goodness, no.
You can make your dog very sick. You can kill it if you give it a high enough worm burden. Okay, don't do that. Not good. Anyway, so how they say here, how does viruses impact the nematobiology, whether they act as drivers or modulators of disease pathogenesis or knowledge gaps? We need to figure this out, right? That's going to be challenging because if you don't have any uninfected worms, it's hard to do the experiments, as we already said. But think about it. We have billions of people infected with these worms, not to talk about the animals, non-human animals. We have billions of people infected. Presumably, they also have the viruses in those parasites. Many of these people have long-term infections, right? So what's going on here? That could be very interesting to study.
Yeah.
Right.
And the viruses could provide an alternative way of targeting these worms.
Maybe. I mean, it seems to me that they're okay with them, right?
Right, but if you can use something in the viral biology to make the virus more pathogenic to the worm.
Yeah. Yeah, I think that would be interesting.
Especially since they infect the reproductive tissue for that second one.
Anyway, Dixon, you see what you missed?
Yeah.
Dixon would have had a lot to say.
Come back, Dixon.
He's not here because he wasn't going to be on TWiV today either. I don't know where he is.
Maybe out fly fishing through the ice? No, you can't really do that.
I don't know where he is. Okay, now Jolene is going to talk about another form of virus discovery.
Yeah, I think there's definitely some overlapping themes here of virus discovery. And
Jolene, did you look at this paper before you made your picks, or you just picked it independently?
I picked it independently. I've been wanting to do this for a while, actually.
Interesting.
So,
by the way, thank you for joining us.
Yeah.
I know you were just here last week, so I appreciate it.
Yes. Well, I'm glad that we're going to talk about this because I think that it brings up a lot of interesting questions about how do we know who the host of a virus is? You know, usually when we talk about it here, we're talking about viruses where it's very clear there's a sequence and maybe we haven't grown the virus in the lab, but we can relate it to other known viruses. And so we have a pretty good idea of what kinds of hosts it's going to have, meaning like it infects mammals, or it'll probably infect birds, or maybe it'll infect many different kinds of vertebrates, or it's an invertebrate virus. But today, the paper that I picked is talking about a virus sequence where we just kind of assumed based on the samples that it came from what the host was, but maybe we were wrong. And
so, Jolene, the previous paper, the transcriptomics doesn't, finding the RNA sequences of these viruses in parasites doesn't mean it's the only host. It could be a host, right?
Yes.
And then finding proteins further supports that the parasites are the host of those viruses, right?
Finding the proteins in the parasite, yes, it does. But I think the difference is that, like, with those kinds of viruses, it's going to replicate in a eukaryote. So usually the host is going to be the biggest thing that's there. And if you see it like those ones we saw, that the fish had signal in different parts of the worm body. They were not living where basically I would expect that if it's a virus of some microbiome component, it would be in the gut, because that's where most of the microbiome is. So if you have that much signal, it's coming from a large population.
Right.
And a lot of, well, for the alpha viruses anyway, not for rabies or I don't know about the other ones, but they're vectored. So just because they infect vertebrates doesn't mean they can't infect invertebrates. They can pass through or infect other host types. But I do think that in that case is pretty clear. I didn't look carefully if they saw the signal intracellularly.
Yeah, I thought the tissue images were pretty incriminating, but that's a good point.
At least the part where it was below the cuticle, there were some places where it was kind of like a maybe sequestered infection.
Yeah.
But the rest of it, it was certainly throughout the tissue. So having I think the key there is having various lines of evidence together. You've got the sequence, you've got the protein. Then you have expression, active expression. All of those things together are good evidence. It would be helpful if they follow up on more of the virus biology.
Right, and the biggest problem here is that complex organisms are full of other things. I mean, we've got our whole microbiome. We're carrying not only bacteria, but fungi and potentially worms. And so if you find something in a human, you don't necessarily just conclude from that, oh, it's a human virus.
Some people would.
Some people
Unless
would,
if it's
and-
highly related to a virus
Yes.
already shown to infect humans, I think it's a reasonable conclusion.
Yeah.
Yes.
Yeah.
Right. If you isolate a novel coronavirus from somebody's lung or nose, that's probably a human virus you've just isolated, or an enterovirus from their intestines.
Mm-hmm. Yeah.
But anyway, this
paper is, before I talk about the paper, actually, I'll tell you what it is. So it's from PNAS in 2023. There are two authors. It's Tianyu Gan and David Wang from Washington University in St. Louis. The title is PicoBirnaviruses Encode Proteins That Are Functional Bacterial License. And so I want to give some background on this. The story is that back in 1988, I believe,
Yep.
there was a human stool sample isolated from someone that was sick, and they sequenced it, and that had this virus sequence. And so they called it picobirnavirus because it's a double-stranded RNA genome in two segments. So it's separated into two segments. And ever since then, many stool samples from humans with gastrointestinal disease and animals have had picobirnaviruses in them. And so that association alone caused us to think that it was a vertebrate virus, at least, probably causing or associated with gastrointestinal disease. It was also shown that picobirnaviruses are associated with this disease called graft versus host disease. So when someone has a transplant, and that transplant involves the cells of the bone marrow or stem cells that can produce immune cells, then there is always the possibility that the individual who received the transplant can be recognized by the transplant cells as foreign, and then the graft will try to attack the new host cells. And that is kind of what we call like rejection, and that's why people who have that might not be exactly what they call it in the medical field, but that's how I understand it. And that's why people who get a transplant need to be matched really close with the donor, and they have to usually be on immunosuppressive drugs to avoid that kind of problem.
Right. And there are two components to that. This is a digression into GVHD, but it's not just that the graft attacks the host attacks the graft.
Mm-hmm. Their picobirnaviruses have also been associated with type 1 diabetes in pregnant women. And so all of these things together kind of coalesced into, we think that picobirnaviruses are human viruses. But that is not the only part of the story. So in addition to them being found in stool samples from humans and animals, picobirnaviruses were also found in respiratory samples from pigs and humans, cattle, and monkeys. So I'm pulling some of this information from a current opinion written by one of the authors of the paper that we're talking about. So this is David Wang wrote this in 2022 in Current Opinion in Virology. And they also have found their sequences of these viruses in horses and deer. They've seen it in blood. All of this together suggested, okay, maybe human virus, maybe not always associated with disease, but strong correlation with disease samples. And the sequences that come out, it's a really small genome overall. The two segmented genomes are usually one segment that's under 3 kb and another segment that is similar in size. So overall, 5 kb or so. They've also found versions of these virus sequences that are just one segment together. And that was it's not totally expected, but it has the same small number of genes. There's an RNA-dependent RNA polymerase, because that's essential for RNA viruses. And there are some other genes of unknown function, and usually a recognizable capsid, but not always. The thing that changed what we were thinking about this virus is that in 2018, there were some papers that came out that showed that the genes of the virus, in front of the gene, there were sequences usually found in bacterial viruses. So this is, I think, maybe something that not all listeners would think about. We talk about viruses that infect people and other eukaryotes, and I talk about phage, the ones that infect bacteria. They're both made of cells, but the cells themselves are different. They have similar processes, they have sort of similar structures, but they're also very different. And the genes that work in a bacterial cell are usually not going to work at all in a eukaryotic cell until you make some modifications so that the eukaryotic cell can recognize them, and vice versa. So the picobirnavirus genes had in front of them sequences that are necessary for translation in bacterial cells. And that was 100% of the genes had these, just like all phages do. That suggested, okay, maybe the host is not a human or another vertebrate. And another study that came out took several of these genes and put them in bacteria and tagged them, and then showed that they could get protein made in bacteria without any other modifications. So that was a good indication that it could make stable proteins. And that's where this paper for today comes in, where they were looking at what else might you do to show that these are infecting bacteria and not something else. Now, there was another hypothesis that maybe they're infecting fungi, but in this paper for today, they're focusing on bacteria.
So, Jolene, has anyone ever looked at a picobirnavirus, like an electron micrograph?
I couldn't find any, but the cool thing that confused me at first is that they have structures of them. And that's because if you express the capsid protein by itself, you can get self-assembly in vitro. Yeah. So they have structures of what is called human picobirnavirus, and also, I think, rabbit picobirnavirus they have structures of.
These are like icosahedral structures,
Yep,
right?
T equals 1.
Oh,
very small.
Cool.
So that's a little unusual to have a structure for a virus which you cannot culture. More often you would have images, electron micrographs.
Hey, a couple of TWiVs ago, we did a paper where they purified a virus from human blood and did the structure.
Yes.
What is it? Was it an adeno-associated virus? They couldn't grow it or something. You got enough out of blood to be able to do the structure.
It's a lot of virus, and that's impressive.
Well, and structural biology has come a ways.
But they did gradients to purify, and you could see bands of virus. There's so much there.
Mm-hmm. Yeah. So to give the kind of quick overview here, there were correlative studies suggesting picobirnaviruses infect humans, but the connection was not clear, and some evidence had been mounting that these picobirnaviruses infect bacteria. So,
and also, you might want to point out that no one ever grew these in human cells.
That's right. Has never been grown in human cells.
If someone had, maybe that would have made a difference. I don't know.
Right.
But they point out in the paper that the failure to grow it is not damning because, for years we couldn't grow noroviruses.
Exactly.
Right.
Yeah. But here they show that there are the presence of some enzymes in the genome of picobirnaviruses that are classic for bacterial viruses. And they're going to look at some potential activity for them in the paper. And like Vincent was saying, without systems to culture the virus in the lab, it's really hard to make a definitive conclusion. I would say we still don't have the answer. And so the thing that they were looking at was the presence of an enzyme that is critical for bacterial viruses to get out of the cell at the end of their infection cycle. These are lysis proteins. So one of the structural differences between bacterial cells and eukaryotic cells is the presence of something called peptidoglycan. This is a peptide and sugar mesh molecule, very large, surrounds the entire cell and gives the bacterial cell its shape. So peptidoglycan is specific to bacteria, and bacteriophages make proteins that degrade that layer so that they can get out of the cell.
And this is something you've worked on before, right?
My lab works on these proteins. They're called endolysins. Yeah. And there's not just enzymes that degrade that peptidoglycan layer. There are also proteins that phage make that can simply block the biosynthesis of peptidoglycan, and eventually the cell will lose its shape and integrity and lyse open and release the phage. So a study had shown that there were a computational study on a whole bunch of metagenome isolated picobirnaviruses, shown that there were some conserved domains in the picobirnavirus's coding sequences that were associated with degradation of peptidoglycan. So I said, aha, this is the enzyme specific to bacterial cells that we should look at. And they took one of these and looked more broadly in other picobirnaviruses and found that there were some more. So this is the M23 family metallopeptidase. So this particular domain is associated with enzymes that remodel peptidoglycan, actually in bacterial cells, also associated with phage enzymes that are used at the exit stage of infection, and associated with phage enzymes that are important in the entry mechanism, because they also have to get their nucleic acid through that layer during entry. And what they decided to do was take this sequence, as well as the related ones that they could find in other picobirnavirus genomes, clone them, and put them into a bacterial cell. And which cell did they choose? Of course, E. coli, because that's where we almost always start. And if you don't have any idea what the host is going to be, it's a good place to start. It turns out that peptidoglycan does have specific modifications in different bacteria, but overall, the structure is relatively similar, and most of these enzymes can work on peptidoglycan from other organisms.
So, Jolene, in a gram-positive, would these work?
Yes. Yes.
The outer layer,
yeah,
doesn't matter.
Well, so in a gram-positive, the peptidoglycan is exposed. So then as long as the enzyme is there, it can work. It's not always that it has the same level of activity, but gram-negatives, which is what E. coli is, have an envelope layer above the peptidoglycan. So if you just added it to the outside, it wouldn't work. But they're expressing it from inside E. coli, which is a little bit problematic, actually. But anyway, they put it on a plasmid, they turn it on, and they're comparing it to a control protein from a bacteriophage called MS2. They're using the MS2L protein, which is actually, we have no idea how it works. We just know that it is the protein required for lysis in MS2. And it does have a toxic effect on E. coli over time. They then see that one of the proteins that they tested was toxic. It kills the cells. And they looked for homologs of those. So there was 3 they tested in the first set, and then they got 5 more from a big marine metagenome study. So even though you might think that these wouldn't work because they are, marine bacteria versus E. coli, which is a human gut bacteria, all 5 of those had activity that were toxic to E. coli. And then they looked at another set of picobirnavirus genomes, what they call conventional. So there are, like I said, some of the picobirnavirus genomes have 2 segments, some have 1 segment, and there are a couple different organizations. So they just picked all the unknown function genes from several that look like they might be this lysine protein. They didn't actually have homology to any conserved domain. They were just like, well, if one picobirnavirus genome has it, maybe another one does too. So they cloned those. They looked for activity. A couple of them did have activity. Out of 16, they got 3 positive hits. They're not all equally toxic. They don't have the same effect. And I think that's because of the way that they're expressing them. So normally, phage have a strategy to get their protein where it needs to be. And here they are just expressing it by itself. So it might not be as efficient at what it usually does.
Right. So what they're doing at this phase is they're kind of walking the taxonomic tree, right? They start with something that none of these picobirnavirus proteins look especially like, strongly like anything else. So they started with couple of candidates that most looked like they might be
Mm-hmm.
license or something, got a hit, then searched homology to those to find other viruses, I'll eventually be able to pronounce that, that are like it, and then pulled those out, and now they're just they're checking to see if this really works the way they think they
Yeah, they're looking in the same place in the genome and they see that they all have a toxic effect on the cell in terms of growth.
So, Jolene, some of these range in amino acids 46 to 311. Are you okay with 46 amino acids being a lysine?
Yes. Yeah, that one I am. 300 over 300 is unusual.
So usually they're smaller?
It depends. So the enzymes that degrade the peptidoglycan are bigger, but the ones like MS2L is relatively small. We don't necessarily know how it works, but the other ones that come from single-stranded RNA phage genomes, they are single peptide that inhibits an enzyme in the host, normally part of the peptidoglycan biosynthesis pathway. So a small protein that can bind to an enzyme and inhibit its function is fine.
They also say none of these have any sequence similarity. So they should have done some AlphaFold, I think, see if they have some structural similarity, right?
They, yes, Dolly would also be good, where they can, so AlphaFold to predict the structure, and then Dolly to look at similarity, like the paper that was done several weeks ago, where they just did like the whole set. The whole virus, there's only a few proteins here. And they may have done that, but definitely that is not discussed here. And I think that one thing you have to keep in mind is that even if you predict structure, that they may not have structural similarity, still have functional similarity. The lysine proteins, oftentimes they just look like an alpha helix. And so you'll get hits, but it'll be to some large helix that's part of another protein.
But this not having a lot of homology or sequence identity, is that common for lysins?
Yeah. Well, for some lysins. So ones, yes. I'll just stop there. The answer is yes. I like to say that because I know this field really well, I'm like, well, they have some similarity, but not all of them. The ones that are not well studied, you get new things all the time. And I'm pretty good at finding lysis proteins in the genome, and I still see new phage genomes, and I can't identify any lysis proteins. So
that's why nobody found this before, because there's no homology to anything, right?
Right.
Okay.
Yeah. So this is positive for toxicity to bacteria. I will caveat this saying that there are eukaryotic proteins that are toxic to bacteria. So they haven't actually shown that this is specific to peptidoglycan. I do think that's a weakness. But what they did next was they looked at the cells that had been exposed to these proteins and looked under the microscope. And then they also stained them. So just looking at them under the microscope, you can see the gross morphology of the cell. And with the stain, this is a stain that can only stain the DNA if the cells are compromised. And they do see that with their control, which is the MS2 protein, they see, actually, was this a phage infection? Yeah, just the protein. And they see that the cells are stained after phage MS2 lysine exposure, and they see that the cells themselves don't look healthy. Let me put it that way. The thing that I would have expected is that they would change shape. It's very common for these proteins to degrade some of the peptidoglycan or prevent its synthesis, and then the cells are no longer rods. And here, that's not what they see. They're also mostly looking way after lysis starts, and the cells tend to degrade over time. So they might not be catching everything at the time that they're looking, but they see that the control, as well as 5 of the ones that they're showing us, have staining, indicating that the cells are compromised. The stain can get in and stain the DNA. So, in summary,
you're okay with that, Julie?
They have toxic proteins. The toxic proteins make the cells lyse or kill the cells. How they're doing that is not clear. though. They really aren't showing that this is specific to peptidoglycan, which is kind of the premise that we need to be confident that this is targeting the bacteria. It could be that it's punching holes in the membrane, and that would be toxic. And bacterial proteins, phage proteins, eukaryotic proteins, they make holes in membranes. You put them in the other host, it still makes a hole in the membrane. So actually, I'm not really convinced. I want to be convinced based on the similarities, but I'm not sure that this is super convincing for the lysis protein.
You don't think the Shine-Delgarno is convincing?
I think that is a better piece of evidence than this.
Did they show that it's a functional Shine-Delgarno? I think you said they put
there
them
was
in
another
bacteria.
paper that put 3 orfs into a cell, and they showed that it made protein by Western.
Yeah, I think the current opinion review that you linked goes into that.
Right.
So if you take like a viroporin, which many eukaryotic viruses have, if you put that in bacteria, it would lyse bacteria?
That's right. Actually, that is one of the definitive experiments most viroporins have to go through to be classified as a viroporin. They both kill, they basically, they clone the gene, put it in E. coli, and then watch the OD go down. Or they clone it, put it in E. coli, and put them on a plate and see that it becomes permeable. So that's exactly what they've done here.
Right.
Okay, so this could be a eukaryotic lysis protein. So the Shine-Delgarno, could it be just accidental?
I don't think so. The thing that I didn't really read that paper, so I can't say for sure, but I would expect that they looked for the classic regulatory sequences found in eukaryotic genes and that they're missing. I didn't see that part, but it definitely has what is expected to be functional in bacteria, to be translated.
And also the Shine-Delgarno sequences are widespread in the Picoviridae.
That's right.
This is a very common feature, which if it was just a spurious thing, you might expect to see them in 1 or 2 viruses, but not conserved.
Another thing, though, is that they talk about in the current opinion that there are some which appear to be using a genetic code used by fungi and invertebrate mitochondria.
Yeah.
So there's also the possibility that these viruses infect fungi. Or that they infect mitochondria, which have similarities with bacteria, a lot of them.
Yes.
So, right. I don't think this is the nail in the coffin that picobirnaviruses are phage.
So you think they are phage, but what would you need for the nail in the coffin?
I think I need to see them cultured. I need to see... You put in the whole genome, I mean, reverse genetics, just put the whole genome into some cells and make phage. Make all the proteins, right? Do the transcription, translation, assembly. That'd be great. Now, finding the exact host is not straightforward. So there are very clever ways now to identify the host of environmental viruses. You could use a probe. You take the samples where these kinds of viruses come from, but don't lyse them all open. Use the probe to stain the cells, FACS sort them so that you can separate out the ones that have signal from the ones that don't, and then do the proximity sequencing. And that way you could enrich for the host of the picobirnavirus. I think that would be a really great piece of evidence that would convince me.
And I think we can conclude from this paper that if you just put the genome of picobirnaviruses into E. coli, you don't get anything because they would have done that.
Yeah, that's right.
Well, now I think that's it sounds simple, but these viruses, when you try to make this may be the case for others as well, but when you try to make the plasmid that has the genome on it and you put it together, it kills the cell right away. So it's really hard to recover a complete reverse genetic system.
Right.
They may or may not have tried that in E. coli, and they may have tried it in E. coli and it didn't work because E. coli is not the normal host.
Right.
So it kills the cell because of the lysis protein?
Yeah. Because
they could knock that out, though, and still get replication, right?
Presumably, yes.
So you
But
can
we
take
don't
out
really
In
know
a
much about the
permissive
replication
cell.
of these viruses,
In a
do we?
permissive cell.
Right, but the problem is you put it in E. coli, the cell dies. So you just knock out the lysis protein and then
If
put it in. You could look for replication in
sure,
the cell by
to
various
make
means.
the reverse genetic system. Yes.
Yeah.
That's right.
You
Now,
could
it may not be
also,
it may not be E. coli because the microbiome of humans has many other bacteria.
Yes.
That's
in it,
right.
many of which are not culturable.
Mm-hmm.
But
What are the levels of these in human stool? Do you have any sense, Jolene?
The PicoBernaviruses?
Is it huge or is it small?
I don't actually know the answer to that. They say that most of the, well, maybe that's I think my reading bias has been, I see that it's associated with diseased stool samples. But there are actually quite a few papers that are analyzing the diversity and the abundance of picobirnaviruses across animals, so that that is probably known.
And from those data, you could probably say, okay, picobernaviruses are found in, I nailed it that time, are found in the presence of this list of microbes
Mm-hmm.
that we know of, and correlate those across and maybe narrow down the list of potential hosts for some of them.
Maybe.
Maybe.
I think there are just too many common.
There are a lot, yes.
The list is huge.
Yeah.
So
It could be more than one host also, right?
Sure.
yeah, there could be.
Hmm.
Doesn't have to just be one kind of host.
Could
You
you
think-
also take the genomes of these picobernaviruses and express them in mammalian cells and see if they do lyse the mammalian cells?
I don't see why not.
Because, I mean, that's a negative experiment, but it would at least get at the argument of, oh, these could be porins or something else that's just cell lethal. It's not
Or
specific to bacteria.
See if they reproduce in those
And see if they
cells,
reproduce
right?
them. Probably that's been tried, right?
I would imagine
I
that
guess
has
so.
been
People
tried.
have tried to culture these viruses in cells. Yeah. All right.
I'm not sure that the reports are not discussed in detail
Right.
in what I read, but I would imagine that would have been an early experiment.
But those experiments would have been looking for replication versus looking for just cell lysis. Right? Maybe.
Maybe they.
I don't know.
Maybe I might imagine they would look for both.
Yeah.
I
Can
mean,
you
you
take,
always are monitoring your tissue culture cells.
Sure. Yeah, if they all round up, then you'd
Yeah.
probably wonder what was going on.
Can you take a sample of microbiome, right? Like actually go in the intestine and pull out a sample and then put it and do some fish.
Yeah.
See if you can find the virus genomes in some bacteria.
Yes. You can. But then you wouldn't know what the bacteria is.
Right.
So the sorting is really important to enrich. Because fish is a low throughput kind of thing
Sure.
for a mixed sample, like a microbiome sample. Although maybe if you did it with more tissue.
Yeah.
Could try the sewage treatment plant.
Well, that's already come out.
I know. That's doesn't.
They do they do some intestinal biopsies on volunteers to get microbiome samples because it's different from what comes out, right?
Well, that's
What's
the
stuck
thing.
on the wall is different from what comes out.
Yeah, most microbiome. Well, and these come from stool samples.
Right.
Yeah.
That doesn't mean that the host is alive in the stool anymore,
Right.
or at least not for very long if it's an anaerobe, which is highly likely if it replicates in the gut, even if it's a fungus, fungal cell.
So you mentioned some animals are shedding picobirnas?
Yes, cattle, monkeys, horses, deer.
How about lab mice?
Bats. I didn't see them mention mice.
That would be convenient, right? Then you could use them.
Right. Pigs.
So, Jolene, here's the question. Do you think anybody's working on this?
David Wang.
I would, yes, I would definitely suspect that David Wang is working on picobernaviruses.
He's a metagenomics guy, right? That's his thing. I'm not sure he's interested in the nitty gritty of bacteria.
Well, if
Maybe.
Tianyu Gan is a postdoc,
That's right.
maybe that's a
Yeah,
name to
but
watch.
on the other hand, he did come up with this idea, so
Yeah.
maybe he wants to pursue it.
Yeah, and
Mhm.
actually, and went and did some lab experiments on it. So
Yeah. So when you go to meetings, Jolene, nobody ever talks about these.
The PicoBernaviruses?
Yeah, because you go to prokaryotic
Oh,
meetings.
yeah. No,
and
they don't
these are
talk
human
about these.
viruses.
No. I don't even remember how I ran across this, but I think it's a really intriguing question. We still don't know what the hosts are. Not even, I mean, we have a very general idea. Let's say
Yeah,
you think
it's
it's
cool.
bacteria. Great. What, not just what bacteria, but how can you figure that out? And I think that many, many metagenomics studies give us new viruses, and we make guesses about who they infect, but being able to make that clear, definitive conclusion isn't always straightforward. And I think that makes it really interesting because that means it's really, really new.
Yeah. And a major reason this even got people's attention is because the I'm screwing it up again.
Yeah.
PicoBernaviruses are so different from everything else that doesn't look like an animal virus. It doesn't look like a phage. This would be, if this is a phage, it'll be an entirely new category of these double-stranded bisegmented RNA phages, which are pretty weird.
Yeah. There are two kinds of RNA phages right now, the single-stranded RNA phages and the three segmented RNA viruses that are also double-stranded RNA, but these are different. And I think that in this case, it's of interest because it's so strongly correlated with human gastrointestinal disease.
Right.
I wanted to tell you an anecdote about RNA phages, right? They were first discovered by Norton Zinder here in New York City, right? He took New York City sewage and screened on E. coli, and he found, I don't know what the first one was, MS2 or some related, Q beta maybe.
Mm-hmm.
But anyway, there's a story that someone wanted to get phage from someone else, and they wouldn't give it to them. So this is a long time ago. They wrote him a letter saying, please send me this phage. And they wrote a letter back because there was no email then saying, sorry, I can't send it to you. But the letter was contaminated with the phage.
Mm-hmm.
So he just put it in
the blender.
Buffer.
Yeah.
You know that story?
I have heard that story. Yeah, it's a good story. I mean, every time that we get calls from people who are experiencing phage in a way that you don't want to, which is you have an outbreak of phage that are preventing you from making protein in your lab. One thing that we tell them is it's in your hair now. So it's all over your skin. You'll never get rid of it.
That's helpful.
So this book, Phage and the Origins of Molecular Biology.
Oh, yeah.
I've had this since I was a grad student. Look, it's all
wow.
It's all broken.
That's your binding?
Yeah, the binding.
That's the binding side. Yeah.
Oh, dear.
The binding just rotted because it's very old, right? But I still kept it because it's a beautiful. It's personal stories about people.
It got a bibliophage on the binding.
Who are working on phages, and it's a collection of stories, basically.
Yeah.
And here I want to just see the Norton Zinder one, RNA phages. Let me see if I can find it to you because it's really cool.
I have a copy of that. I think I have a copy of that book. Phage, yeah, but mine is the expanded edition, actually.
Look at that.
It does have a binding. I read it.
I did too. It's really good.
Mm-hmm.
Really like it very much. Where the hell is Norton Zinder? Here. Maybe I'm thinking of another book. Probably it's not in.
There's
this
also
one.
this one that has a lot of stories, The Molecular Biology of Bacterial Viruses by Gunther Stint.
Yeah, that might be
that
it.
one has a lot of stories in it.
Anyway, I may be one of these two books has the letter story in it, you know.
Mm. So there you go, PicoBernaviruses.
Cool. PicoBernaviruses, yeah.
I will never look at them the same way.
I'm very curious, and I really hope that it gets worked out. And I think that there's interesting experiments to be done.
Yeah. I think we can do one round of emails. What do you think?
Sure.
Jolene, can you take that first one?
Yes. The first email is by April. Dear TWiV team, first let me say how much I appreciate everything you do to make virology accessible and engaging. TWiV has deepened my understanding of science in ways I never expected and made listening to in-depth discussions of topics I don't understand fun. That brings me to a suggestion about something that comes up occasionally on the show. Every once in a while, you get an email asking you to make the content more accessible for non-scientist listeners. When you address this, it sometimes feels like the response is to overcompensate, defining every method, every piece of jargon, and diving into granular detail. While I understand the intent, this approach can get a bit tedious, and for listeners like me, it doesn't help as much as you might think. I'm not a scientist. I don't have an advanced degree in science, and it will take more than a few definitions to get me to understand what you're talking about. While I appreciate the effort, these details still go way over my head. Sometimes you even have to explain things to each other, so I definitely will not understand the technicalities. Instead, I have a suggestion. At the end of each paper discussion, could you set aside a little time for a lay summary? Similar to what NIH asks for in grants, this would be a brief explanation of what was the research question? How did they try to answer it? Not in detail, just broadly. Did they study cells, mice, humans? What did they find? What does it all mean? A so what section. Why does this matter and what are the broader implications? I think this approach would help non-scientist listeners like me stay connected to the bigger picture without interrupting the flow of your conversations. It could also be a great way to highlight those moments where Brianne shares what the findings, quote, make her think about. Alan might be particularly great at summarizing for a lay audience, and Rich already does a good job of remembering we don't all have the same background as you all do. This would supplement the in-depth discussions and would be a way to tie together everything in more accessible language at the end of each paper. To make this a little easier, I've attached 2 prompts that you could put into ChatGPT after uploading an article. The first will give you a lay summary, similar to the way Vincent sometimes summarizes things he's trying to understand on some of the other podcasts. The second will give you a really simple summary suitable for Congress. I don't know which you would want to use, but it seems like it would be fun to discuss where you agree with or disagree with ChatGPT's summary. Thank you so much for considering this suggestion. I've learned more from your show than I can put into words, and I'll continue listening even though I can't quite follow the details. Sincerely, April.
Mm-hmm.
April, I think that is a valid point, that when we have to explain it to each other, very likely that the non-scientist listeners who we do want to reach with the main message of the papers might be a little lost. So kind of zooming out, the way that we suggest to our trainees when they're giving talks or when we are giving talks, even to broader scientist audiences, is a good idea.
Like I ask you questions many times today.
Mm-hmm.
Maybe it's most people don't understand the answers, right?
Yeah.
So I ask you, can you put a viroporin in blah, blah, blah. People don't know what a viroporin is.
Right. Mm. Yeah.
It occurs to me that we do attempt to do this. We're just not very consistent with it. Kathy is particularly good about saying, wait, before we get into the paper, let me give the top line summary. So check out episodes with Kathy on them. And some of the others of us will also sometimes remember to do this. Rich is very good about top line. What is this virus? It's naked icosahedral RNA virus, something like that. But yeah, we like these reminders to do this.
Should it be before the discussion?
I think it ought to be before. Yeah,
she suggests after. Yeah.
I think before is probably the place to do it and put things into context and say, these the picobirnaviruses were found in humans and they may not be a human virus, and this is what we're going to talk about, kind of thing. Which we kind of did. Jolene, actually, I think,
I tried.
From where I was sitting, did a nice job of leading into this.
So I think it should be limited. Sometimes Kathy goes on a bit,
The problem is how much detail do you include? Yes.
Yeah.
We all go on.
Yes.
Yeah. But if in the summary, you shouldn't go on because
right.
Sometimes I feel, well, we don't have to do the paper now.
Right.
It's already summarized. But we should give it a limit, because I'm thinking then I could pull it out and make a short video for people on top of it, right?
Vincent, you're like my
1 to 2 minutes.
You're like my students. I tell them there don't need to be surprises. They're like, I don't want to give away the punchline. I'm like, tell them the punchline.
Spoilers
They'll
are
get
okay
it
in
better
this.
if you do.
Yeah. I don't mind the punchline. It's just
The juror dies in the end.
Oh,
if
Yeah.
you go through all the details, you don't want to repeat it. But I'm going to take these points and put them in future show notes, and
It's a good idea.
we can. I mean, do you think 4 different people should do this? There's four bullet points, 4 different people, or one
I think
person?
one person, whoever's leading with the paper, should probably just be reminded to do a 1-minute top line summary.
Or less.
What are we going to be talking about? And if it's a really hairy paper, like, going way into the molecular weeds, just zoom back out and say, what is the point? What are we going to dig into the weeds to try to figure out? What's the overlying phenomenon?
The miniest mini abstract. No, it's really like she said, what the NIH asks for.
Yeah.
A lay summary.
I hate writing those.
Yeah. They don't really address what you're doing most of the time.
Right.
Alan, could you take the next one?
Sure. Rachel writes, in your discussion of the first paper, the snippet, in TWiV 1167, the mention of proteases and protease inhibitors had me thinking, this sounds very familiar. Dixon then brought up that there is a condition that people have involving a lack of protease inhibitor, and I believe he may have been thinking of alpha-1 antitrypsin deficiency, a condition that I have. Dixon wasn't sure how this condition affects people, and the answer is, it depends on their genotype, but it also depends on environmental factors and possibly other not yet understood factors as well. To briefly explain it, alpha-1 antitrypsin is made in the liver, so people with the condition can be liver affected. The protein is made in the liver, but it is misfolded and can't get out of the liver, which causes liver scarring. This scarring can cause anything from mild disease to liver failure. A liver transplant cures the condition, as long as the donor doesn't have alpha-1 antitrypsin deficiency. Apparently, the most important role A1AT plays is in the lungs, so alphas, as we call ourselves, can be lung affected as well. It can mean a high frequency of exacerbations, i.e. lung infections, as well as other conditions such as COPD. Most alphas are one or the other. Some, like me, are only mildly affected in both the liver and the lungs, so far anyway, despite having a severe genotype, ZZ. Many lung-affected alphas receive weekly infusions of plasma containing A1AT. This replacement therapy is believed to help preserve lung function. Many lung-affected alphas progress to the point of needing a lung transplant. These people will then have well-functioning lungs, but will continue to lack A1AT, so many of these people resume infusions sometime after their transplant. The Alpha-1 Foundation is a fantastic resource for those of us who are affected, as well as those who just want to know more. There are a handful of other conditions associated with A1AT deficiency. I also have a kidney transplant, the need for which resulted from a post-strep infection of my kidneys as a child. This was treated to stop the acute infection, but my kidney function gradually and slowly declined until I received a transplant 50 years later. I have asked Alpha-1 researchers, could there be a connection between A1AT deficiency and my inability to fully recover from post-strep glomerulonephritis? They say they would need a biopsy to biopsy my native kidneys to find out. I suspect that a savvy team of immunologists might be able to come up with some interesting thoughts about this. I have often thought that I would love for your team to devote a whole episode to this condition. Wouldn't that be a great topic for Immune? I love all your shows. Thank you for all that you do. Rachel, who is a retired high school teacher of chemistry and biology and all-around lover of all things science. That's really interesting. I'm not at all familiar with this condition, but it sounds like a peculiar set of outcomes, and I don't have any insights on the immunological aspects.
I'm not sure what the best podcast would be to talk about it, right? These things are not, as far as I can see, connected to immunology. I do think there's some gene therapy going on to try and replace alpha-1 A1AT, with a vector. So this is amenable to gene therapy.
Right.
It's a monogenic defect. Yeah. Interesting. Thank you. These are great.
I think she's also asking about whether her transplant has something to do with the.
Right, whether her kidney failure after strep, I mean, an immune episode
I
on
see.
immune
Mhm.
responses to, yeah, I guess that's really the focus that she would ask for on immune.
Yeah.
To what extent could a deficiency in this enzyme affect your ability to respond to, say, a strep infection?
Okay, got it. All right, Lee writes, hey, TWiV, I'm pretty concerned about the H5N1 levels in wastewater in California. And now the Canadian in ICU with confirmed H5N1 also having COVID. Feels y'all are on a very slippery slope. Why do you say you? It was you too.
We all would be, yep.
Should I be buying in more masks? Feels like we are a winter flu season away from a new pandemic. All right. Listen, Lee, since we're paying so much attention to H5N1, it's not going to become a pandemic.
You
know,
get
all
the pandemic you don't expect.
Yeah, like SARS-CoV-2, boom, we weren't looking for it. H1N1 in 2009 came from nowhere, right?
2024 Congo mystery disease.
Now, the thing is, Lee, nobody can predict if we're going to have an H5N1 pandemic. If anyone does, they're lying. You can't. We have no idea. We need to be prepared.
And I would add that we are always a winter flu season away from a new pandemic and have been for centuries.
There's going to be a flu pandemic next. I think that's
Sure.
next.
Sure. I was expecting a flu pandemic before we had COVID-19. I thought, yeah, next pandemic, it's obviously going to be flu.
We're going
But
to
we
have
have
to
to be
pin it
prepared
on
for
you,
each
Alan. You were expecting it. That means you must have caused
I was
it.
expecting it, therefore it didn't happen.
We have to be prepared, and I think we are pretty much prepared. As long as we can continue to do infectious disease research, we'll be prepared here in the U.S. Okay.
And distributing, maintaining the vaccine distribution system, because we can generate a new flu vaccine much faster than we generated a COVID-19 vaccine.
The group of people who select the strains that goes into the next vaccine, where are they? An independent group?
Yeah, WHO has them, and they're from all over the world.
Okay.
Yeah, that's
they're
an
not
international
US
group.
based.
They are not going to be killed by mismanagement of HHS.
No.
At least not immediately.
So the other countries can continue to function scientifically.
Yeah.
Okay, let's do some
ASV
picks.
is going to be in Canada. So, just a reminder.
So the end of Lee's email here, I was curious about it. He signs off, or she signs off.
Kia
ora rawa.
It means thank you very much.
Yeah, in Maori. Okay.
It's time for some picks. Let's do some.
Maybe that's what that means by y'all are on a very slippery slope. If you're in New Zealand, you may be a little smug about pandemics.
Yeah, maybe.
Okay.
Yeah, because they're not going to get hit. They're just going to shut their borders.
And their seasons are inverted from ours, so they'd probably do even better than they did in COVID-19.
Yeah. Okay. Now I get the y'all.
Okay.
Okay. Picks of the week, Alan. What do you have for us?
I have a site that I've been enjoying for almost a year now. It's called SondHub, SONDEHUB, and it is a tracking site for radio sondes on weather balloons. Okay, so this kind of remotely feeds into the weather arc on TWiV, but I discovered this through ham radio. Twice a day at 0 hours and 12 hours UTC, weather stations around the world. There's one in Albany. There's one in one on Long Island. They're just and then all around the world. Every country has these. In every region where they've got a weather service, they launch weather balloons, and these are latex balloons that rise up all the way through the atmospheric layers. They go up to about 100,000 feet, like 30,000 meters. And as they go, they're sealed balloons, and they expand according to, gas law. And then at a low enough pressure, way up at the 100,000 foot level, they burst, and the payload on them falls back to Earth. The payload is a little device called a radiosonde that has a tiny little radio transmitter, a thermometer, a hygrometer, and a barometer, and a GPS. And it uses a commercially off-the-shelf board, circuit board. It's got a little styrofoam box around everything. I should have brought one up here to show off. But this whole thing, it's got a little parachute so that as it comes back down to Earth, it won't hurt anybody as it hits. It weighs about as much as a coffee cup. And the Weather Service considers these disposable because they land dozens or sometimes, 100 miles from where they were launched, and they're inevitably coming down in the ocean or in the trees or some field or something. They're not going to go out there and get these devices, but they buy them in bulk, and they only cost a few bucks a piece. So some ham radio operators about 5 years ago started monitoring these because you can receive the frequencies and you can actually watch the weather balloons in real time and see them go up. And tracking on the GPS, it gives the wind, right? And this is what the Weather Service is doing. They're putting all these data into the global weather models. But the fun thing is that when the balloon bursts and the sonde comes down, it's going to be sitting out there somewhere as like a treasure hunt challenge. Right? Because it's going to land in some random location on the map. And because there are now geeks like me, ham radio operators, I don't actually have one of these receiving stations yet, but one of my neighbors does, who's a ham. You can track them almost down to Earth and then just go out, and if it's someplace accessible, you can pick these things up and reuse the little circuit board in them for your own projects. And it's just a fun challenge to watch these balloons. And, you can open up this site in the evening here on the East Coast when they've done the 0 UTC launch is about 7 p.m. here on the East Coast. So, I'll pop up the site and I'll see the balloon from Albany is now over Pittsfield or something and ascending, and I can watch it go. And every now and then one comes down in the western Massachusetts area. And I've recovered a couple of these. And they're just, you go out and find in some park or field or something, there's just this random object that just fell from the edge of space. And
so you use your receiver to keep monitoring the signal when you're close.
Yes. And this is, there's a site that tracks air traffic in a similar way with just people, ordinary citizens who have a little Raspberry Pi, usually with a radio receiver USB attached to it. And then they just monitor the signals. And so these are on the SondHub site, you can see little green dots all over the place. And those are individuals who've put one of these things in their house, and that receives. And because these things are up, 70, 80,000 feet, the signal covers a very wide area and you can receive it easily down on the ground stations. And I'm also linking to a YouTube video that describes this whole process very well. That's from a Linux conference in Australia. So something to geek out on.
Cool. Jolene, what do you have for us?
Yes, I picked a book. It's called 10 to 25, The Science of Motivating Young People. It's written by David Yeager, who is a psychologist at the University of Texas in Austin. And I heard him give an interview on our local public radio, or Texas Public Radio, right before it came out. I read the book. It's very interesting. So he takes this approach to explaining basically why teenagers act the way they do and how you can interact with them in a way to motivate them to be productive and honest and helpful and to reach their full potential, because young people have a lot of potential. So he talks about how it's been shown that around 10 years old, puberty changes the brain, and young people go from being wonderful children to individuals who crave socially rewarding experiences. And he links this to an evolutionary past where at that age, you needed to start to establish your position in society and earn respect. And so you would shun humiliation and shame. And much of the ways that we interact with young people assume that they are stupid, and kids these days just don't do anything right. But he puts together the kind of best practices mentor framework from many, many different fields. So parenting, teaching, coaching, manufacturing, all kinds of areas. And he pulls that all together into this framework of how you can interact with young people in a way that will validate them so that they feel they can earn respect, but also give them opportunities and space to grow. And this does not mean holding them to lower standards, but holding them to very high standards. So I thought it was a really interesting read, and I listened to the book, but I've also bought the book because I think I want to try to apply a lot of these principles.
Very interesting. I don't disrespect young people at all. I think they're just as part of society as everyone else.
I do sometimes make cracks about different generations, but I do that to boomers as much as I do to zoomers. So.
All right. My pick is an article in Science-Based Medicine by Jonathan Howard. And the title is funny. It's Dr. Vinay Prasad, who you may have heard of. Bloody pundit who knows nothing, apparently. Sabotaging RFK Jr.'s confirmation will increase vaccine hesitancy. And doctors criticizing RFK Jr. paved the way for his ascendancy. So here's the story. I can forgive you for thinking that my article is satire. It is not. Dr. Prasad actually published articles with these two titles that are the title of his article. In one of them, the sabotaging RFK's confirmation will increase vaccine hesitancy. Dr. Prasad argued the best way to curb vaccine hesitancy is to approve him and redirect his energy to generating more data to answer key questions that remain unanswered. Which childhood immunization program is optimal? And to which the author, Jonathan, says, it's hard to know where to begin with something this asinine. According to Dr. Prasad, data about routine vaccines is lacking. Anti-vaxxers need to change their minds is more data. A cluster RCT of the routine vaccine schedule is both feasible, ethical, and a good use of resources. The ideal person to generate this data is a crank who likened vaccines to the Holocaust and already has a body count of 83 in Samoa. Keeping RFK Jr. from office would empower anti-vaxxers more than giving him power. I won't insult Dr. Prasad by pretending he actually believes any of this. He's not even trying to make coherent points anymore. It's just bad faith, performative argumentation, and nothing else.
Yeah.
And I love that last sentence. That's what all these people do. They don't even try to make coherent points anymore. Prasad is on record.
As I said at the top, this is shock television. This is about getting you to look.
It's anyway, the rest of the article talks about the other points of Prasad. And he's on record saying that RFK Jr. has more reliable information than Peter Hotez about COVID. This is just nonsense, right? It's asinine. It's asinine. My gosh, this is what we've ended up arriving at in this world, in this country. I don't know about the rest of the world. Anyway, if you want to get frustrated, read it. We have two listener picks. One from Sil. How many kids might this little gem entice into science? This is a NPR Goats and Soda post by Kamala Thiagarjan. A fold paper insert lens. This $2 microscope changes how kids see the world. It's an article about Foldscope, which is this paper microscope, very cheap. So that's pretty cool. And Jennifer, hello, Doctors TWiV. I have a pick for you, an app called Minutia, the anti-social media app. Minutia.app.org. It's an anonymous and anti-social media app. No comments, no likes, no profiles. Once a day, at random moments, everyone receives a notification, and you capture whatever is in front of you in that moment. No selfies, just pictures of ordinary life. Then you get to scroll the moments of one random stranger for a minute or so. It's been really fun to just document daily life, and I think it's something some of you, maybe not Vincent, will enjoy. Sincerely, Jen. Yeah, I'm not sure I would enjoy that, but how would you know that, Jen? How would you know that, Jen? Ha ha. Ha ha. That's TWiV 1175. Show notes at microbe.tv/TWiV. Questions, comments, picks, TWiV at microbe.tv. And if you enjoy these programs, please support them. Microbe.tv/contribute. Alan Dove is at turbidplaque.com, alandove.com. Thank you, Alan.
Thank you. It's always a pleasure.
Jolene Ramsey is at Texas A&M University, ramseylab.vercel.app. Thank you, Jolene.
Thanks. This was great.
I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV. Ronald Jenkins for the music, and Jolene for the timestamps. I got confused because Jolene is sitting right there.
Sorry.
It's not your fault.
No, not your fault at all.
You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.